Welcome dear viewers. This video is all about the Herpesviridae family of viruses. I am Dr.M.Manigandan, Assistant Professor, Department of Microbiology, Sacred College, Tirupattur. Introduction. The herpes viruses are an important group of large deoxyribonucleic acid enveloped viruses with the following features in common. That is virion morphology, basic mode of replication, and capacity to establish latent and recurrent infections. The human herpes viruses are Herpes Simplex virus 1 and 2, Varicella zoster virus, Epstein-Barr virus, cytomegaloviruses. These viruses are ubiquitous in nature. Cell-mediated immunity is important for causing symptoms and controlling infection with these viruses. Herpes viruses encode proteins and enzymes that facilitates the replication and interaction of these viruses within the host. Epstein-Barr viruses and human herpes viruses are associated with human cancer. Structurally, these herpes viruses are large enveloped viruses that contain double-stranded DNA as their genetic material. The virion size is approximately 150 nanometer in diameter. Herpes viral genomes are linear double-stranded DNA, but they differ in size and gene orientation. The DNA core is surrounded by an icosahedral capsid containing 162 capsomeres. This capsid is enclosed by a glycoprotein containing envelope. Herpes viruses encode several glycoproteins for viral attachment, fusion, and for escaping immune control. Attached to the capsid and in the space between the envelope and the capsid is called as a tegument that contains viral proteins and enzymes that helps initiate replication. As enveloped viruses, the herpes viruses are sensitive to acids, solvents, detergents, and drying. Mode of transmission of herpes virus family. This transmission may vary according to each viruses. The HSV type 1 virus transmitted primarily by saliva and close contact. HSV type 2 transmitted by sexual contact. Varicella zoster virus transmitted majorly through respiratory and close contact. The cytomegalovirus also majorly transmitted by close contact, transfusion of blood and blood products, tissue transplant, and through congenital transmission. Epstein-Barr virus acquired by sharing saliva, that is by kissing. In this case, also it transmits from sharing of water bottles. Replication of the Herpes simplex virus type 1 and 2. Replication initiated by the interaction of viral glycoproteins with cell surface receptors, and virus fuse its envelope with the plasma membrane, releasing the nucleocapsid into the cytoplasm. The enzymes and transcription factors are carried into the cell in tegument of the virion. The nucleocapsid docks with the nuclear membrane and delivers the viral genome into the nucleus, where the genome is transcribed and replicated. Transcription of the viral genome and viral protein synthesis by three phases. First one is the immediate early proteins, consisting of proteins important for the regulation of gene transcription and takeover of the cell. The second one is the early proteins, consisting of more transcription factors and enzymes, including the DNA polymerase. Late proteins, consisting of mainly of structural proteins, which are generated after viral genome replication has begun. The viral encoded DNA polymerase, which is a target of antiviral drugs, replicates the viral genome. Viral encoded scavenging enzymes provide deoxyribonucleotide substrates for the polymerase. The polymerase and other viral enzymes facilitate replication of the virus in non-growing cells that lack sufficient deoxyribonucleotides and enzymes for viral DNA synthesis. Example, neurons. Other proteins manipulate cellular machinery to optimize replication inhibit immune responses, and inhibit apoptosis or establish latency in the host cell. The capsids now reach the nucleus, fills the DNA of virus that is replicated inside the host nucleus, and acquire an envelope at the nuclear membrane or Golgi membrane, then exits the cell by exocytosis or by lysis of the host cell. The transcription, protein synthesis, glycoprotein processing, and exocytotic release from the cell are performed by cellular machinery. Viral latency. The viral genome is transcribed by the cellular DNA-dependent RNA polymerase enzymes and is regulated by the viral encoded enzymes and cellular nuclear factors. The interplay between these factors determines whether a virus to undergo a lytic life cycle or a persistent infection or a latent infection in human host. Cells that promote latent infection transcribes a special set of viral genes without viral genome replication. Progression to early and late gene expression results in host cell death and causes lytic infection. HSV was the first human herpes virus to be recognized. The name herpes is derived from a Greek word meaning to creep. It causes cold sores. These were described in antiquity, and their viral etiology was established in 1919. Pathogenesis of HSV type 1 and 2. The mechanism involved in the pathogenesis of HSV type 1 and 2 are very similar. The virus generally causes lytic infections of fibroblasts and epithelial cells and latent infections of neurons. Both viruses initially infect, replicate in mucoepithelial cells, cause diseases at the site of infection. HSV viruses binds quickly and efficiently to cells through an initial interaction with the heparan sulfate, a proteoglycan found on the outside of many cell types, and then through a tighter interaction with receptor proteins at the cell surface. Penetration into the cell requires interaction with nectin-1, that is herpes virus entry mediators type C, an intercellular adhesion molecule that is a member of the immunoglobulin protein and similar to the poliovirus receptor. Nectin-1 is found on most cells and neurons, and then these viruses establish latent infection at the innervating neurons. HSV1 is usually associated with infections above the waist and HSV2 associated with the infections below the waist. HSV1 and 2 are common human pathogens that can cause painful but benign manifestations and recurrent diseases. The virus initiates infection in mucoepithelial cells or breaks in the skin. The virus replicates in the cells at the base of the lesion and infects the innervating neuron, traveling by retrograde transport to the ganglion, that is the trigeminal ganglia for oral HSV and the sacral ganglia for genital HSV. Upon reactivation, the virus then returns to the initial site of infection, and the infection may be inapparent or may produce vesicular lesions. The vesical fluid contains infectious virions. Recurrent facial herpes infections are generally activated from the trigeminal ganglia. Both HSV virus 1 and 2 can cause significant morbidity and mortality on infection of the eye or brain and on disseminated infections of an immunosuppressed person or a neonate. Infected people may experience recurrent mucocutaneous HSV infection, that is cold sores, fever, blisters. Several infections were caused by Herpes simplex virus 1. The first one is the gingivostomatitis. This one occurs primarily in children and is characterized by fever, irritability, and vesicular lesions in the mouth. The primary disease is more severe and lasts longer than recurrent infections. The lesions heal spontaneously within 2 to 3 weeks. Many children have asymptomatic primary infections. Herpes pharyngitis is becoming a prevalent diagnosis in young adults with sore throats. The second one is the herpes labialis. It causes fever, blisters, or cold sores which are milder, and these are all recurrent form of infection. This herpes labialis is characterized by crops of vesicles, usually at the mucocutaneous junction of the lips or nose. In this infection, recurrences occurs more frequently and reappears at the same site. The lesions of herpes labialis or gingivous stomatitis begin as clear vesicles that rapidly ulcerate. The vesicles may be widely distributed around or throughout the mouth, involving the palate, pharynx, gingivae, buccal mucosa, and tongue. Many other conditions were caused by Coxsackie virus lesions, canker sores, and acne resembles with the HSV lesions. The third infection is the keratoconjunctivitis. The infection is characterized by corneal ulcers and lesions of the conjunctival epithelium of eye. The recurrent infections can lead to permanent scarring, corneal damage, and blindness. Encephalitis, the fourth infection, which is characterized by necrotic lesions in one temporal lobe. It causes fever, headache, vomiting, even seizures, and altered mental status. Herpetic whitlow, that is a fifth infection, which causes pustular lesions of the skin of the finger or hand. It can occur in medical personnel as a result of contact with the patient's lesions transmitted through cuts or abrasions. In HSV type 2, there are several infections including the genital herpes and encephalitis. In genital herpes, there is a more painful vesicular lesions of the male and female genitals and also in the anal area. The lesions are more severe and protracted in primary disease than in recurrences. Primary infections are associated with fever and inguinal adenopathy, which are frequently accompanied by itching and a mucoidal discharge with severe pain. The asymptomatic infections may also involve due to the source of infection of other healthy individual. In men, this virus causes infection in the glands or shaft of the penis and also in prostate, occasionally in urethra. In women, it causes infection in cervix, vulva, vagina, perianal area, or inner thigh. The neonatal herpes, it is a devastating and often fatal disease caused most oftenly by HSV type 2 virus. It originates chiefly from contact with vesicular lesions within the birth canal. It is possibly infects at the baby's scalp because of the because the mother is shedding this herpes virus at the time of delivery, or the virus is acquired postnatally from family members or hospital personnel. The infection varies from severe disease, that is disseminated lesions or encephalitis, to milder local lesions, that is over the skin, eye, or mouth, to asymptomatic infection. The encephalitis may be developed due to the cell-mediated immune response not developed in the neonate at the birth. The HSV disseminate to the various sites like liver, lung, and other organs of the neonate, as well as to the central nervous system. Progression of the infection to the central nervous system may result in death, mental retardation, or neurologic abnormalities, even with treatment. The drugs of choice for the treatment of herpes simplex virus 1 and 2 is based on the prototype acyclovir. It is the most prescribed anti-HSV drug, followed by valacyclovir, the valyl ester of acyclovir. Next is the pencyclovir and famciclovir, a derivative of pencyclovir. These are related to acyclovir in their mechanism of action, but have different pharmacological properties. No vaccine is available up to the date, and I thank you very much for your patient listening. Thank you.